As Christine mentioned, I'm the chair of the Scientific Advisory Board, which I'm very honored to participate in. It's an outstanding group. And so my first order of business is just to give you a very brief update on what that board has been doing. So our biggest job is to review the applications that we receive for your money for research. And we do formal reviews and score the applications, and then the final funding decisions actually get made by your board of directors, but we give the scientific overview. Obviously, I didn't test it. There we go. So before I start, and I'm going to talk about Pegvaliase, so I have to tell you, disclose up front that I've been working with any company that will try to do a new therapy for PKU, to be honest. So I have to give that disclosure. Whoops. So this is my agenda for the whole morning, actually, this first hour. So I'm going to do a very brief overview of what we're doing for the SAB. I'm going to give an update on the Pegvaliase trials. Dr. Mark Toohey from Trayer Therapeutics is going to talk about what they've done with Catherine Durr's probiotic. I'm going to give a bit of an update on our gene therapy research, but we're nowhere near the clinic yet, so I'm going to keep that brief. And then finally, Dr. Tom Franklin will talk about the progress in the home Phe monitor challenge. So this is just a list of the projects that were funded in 2016, and you'll see a bit of a conflict on there because I also got some funding, which I'm very grateful for. And our work is, again, to do the gene therapy. You've heard from a couple of these people, and we'll hear from others tomorrow about the progress. And we had a meeting on Thursday, before a lot of you got here, where most of the scientific advisors were here, and we also had quite a few of the people that are being funded come and give us more detailed scientific presentations, and then we could grill them extensively about their work. And it's the first time that we've done that. I thought it was extremely productive and useful discussion. And then the final part of the day, what we discussed was where we might like to invest in the future. And one of the big topics that we've talked about is to try to develop a bigger study of outcomes in all ages of patients, and so that's on a very, very early formative stage, so I don't really have any details to give about that yet, but that was one of the big topics of discussion. So let's switch to the Pegvaliase trial. So you all know what PKU is. It's deficiency of this enzyme, phenylalanine hydroxylase, and people have tried to use this enzyme as a therapeutic, but for a variety of reasons, it just does not play very well in the test tube. So it's very difficult to purify it in any amount that you might be able to try to give back to a person. So that's kind of gone by the wayside a little bit, although there are a few labs that are still trying to do this. The important thing about PKU is, although the enzyme's missing from the liver doesn't really care. The problem is that the phenylalanine goes up, and even though it's a normal amino acid at very high levels, it does something to the brain. And there's lots of potential theories and some data on what it does to the brain, and that's one of the hot topics of research in the animal models. And there's probably some truth to all of these potential possibilities that are on the right-hand side of the slide. But what we do know is it's all related to the blood phenylalanine, or really the brain phenylalanine, which is related to the blood phenylalanine. So whatever we can do to get the blood Phe down and consequently the brain Phe down should really treat this disease. And so there are many, many different potential ways that this could be done, and some of them have done in practice and some of them are theoretical. The thing that, of course, is done in most patients now is dietary Phe restriction. And what we're going to talk about today is subcutaneous phenylalanine ammonia lyase injection. And then later on, I'm going to talk about liver-directed gene therapy. So just those two possible approaches. So Pegvaliase is an enzyme substitution. So it's not enzyme replacement. Replacement would be putting phenylalanine hydroxylase back, but you've already been introduced to this molecule by Dr. Durr yesterday. She's put this enzyme into bacteria and are feeding it to the PKU mice. This is different. This is taking the enzyme, putting it in a syringe and injecting it under the skin. And so the subjects get an injection once a day of the Pegvaliase enzyme. And it, as you learned yesterday, takes phenylalanine, cuts off an ammonia and leaves tranexamic acid. In your body, it rapidly excretes both of these molecules. So that enzyme, there's many bacteria that produce it and use it for this reaction. It's a foreign protein, so it had to be coated with polyethylene glycol, or PEG. So some people still call this PegPal. The brand name or the formal name is Pegvaliase. And I want to point out that Dr. Ray Stevens, who was at the Scripps Institute with NPKUA money, was really the lab that came up with the final product, trying all different species of the enzyme and all different ways of pegylating before they finally came up with the drug. Christina Sarkissian, who's a scientist at McGill University in Montreal, first showed in the PKU mice that this stuff worked, and then BioMarin has taken it on, and over the past 8 years we've been through multiple clinical trials to try to ultimately get FDA approval. We are very close to the finish line. The disappointing thing I'm going to tell you right now is that I can't give you the punchline, and it's because we're very close to the finish line. There is a federal law that if we present unpublished data to families, that's considered marketing of an experimental drug and it's illegal. So I'm going to describe the study to you. Many of you have probably seen some of this before. And then I'm going to give you a website link where actually the data are published, but I can't publicly tell you, so it's disappointing, but I can't give you the punchline. But I'll tell you about how the studies were done. So phase 1, which started 8 years ago now, was simply for safety. 25 adults with PKU got a single shot of PegPal. I'm going to always say that. I have a hard time remembering the switch. And just to look for safety, and we learned in that at the highest dose we gave, the blood fees dropped. That information is published, so I'm not hopefully going to go to jail for telling you that. The blood fees dropped, but some of the subjects got skin rashes and some of them joint pains when they developed antibodies against the drug. So we knew that was a possibility, and it's something that we've learned to deal with. After that, we moved into phase 2 trials, and there have been 3 of them. And I think of this as sort of the Goldilocks experiment. So PAL-002, we started with very low doses, and we went up very slowly to try to make as few of reactions against the drug as possible. And that was basically the too slow version. In PAL-004, we tried to start people at a high dose right away, and everybody got sick, so that was the too fast version. And then 165-205 was just right. So it was basically somewhere in between, sort of balancing safety versus getting someone's blood phenylalanine down. Most of the subjects that have participated in those phase 2, in those early phase 2s, have moved into a long-term extension and are still on drug, and we have people that have been on PegPal now for 8 years. The critical trial, which was what are now presenting to the FDA, are PRISM 1 and PRISM 2, the phase 3 trials. And PRISM 1 is for people who really hadn't previously been in any trial, had not seen Pegvaliase at all. And if they got to a dose that worked for them, that their blood fee dropped safely, they were eligible to move into PRISM 2, which has this critical step in it, this randomized discontinuation trial, where actually some of the people went on placebo shots for 8 weeks. And the goal was to compare what happened to the people on the placebo shots versus the ones that stayed on the active drug. That data was just unblinded in March, and I had the privilege of presenting that data then in Florida at the Society for Inherited Metabolic Disorders meetings. So for the phase 3 studies, these were the people that were eligible. They were adults with PKU. You had to have a high blood fee. And you had to, we collected diet information, but we did not control the diets, but they had to be willing to kind of basically keep their diet the same and not make big changes in it. As you'll see in a minute, most people were not really on any dietary restriction. We only had about 10 out of 200 and some that were using any formula at all, and patients couldn't be on Kuvan. So this was PRISM 1. At the beginning, they were all randomized to either get 20 or 40 milligrams. I want you to understand that 20 milligrams works for about half the population, 40 milligrams for a few more. So if you got randomized to 20, you might not get an effect. So those people, if they hadn't had a response to 20 milligrams, wouldn't go into the PRISM 2. But later on, as you'll see, they had an opportunity to get more drug later and have an effect from that. So they were randomized to 1 and 2 doses, and they went for up to 37 weeks to try to see if this worked for their blood Phe. And if it did reduce their blood Phe to some, it only had to come down 20%, then they were eligible for PRISM 2. They stayed on their same dose, but some of them went on placebo for this 8 weeks. Then after that, there was a period where everybody, where we got a lot of blood levels to look at levels of the drug in the blood. And then ultimately, everybody moved into an extension trial where people were able to adjust their dose up or down from as low as, some people are on even like 5 milligrams a day or lower, and up to 60 milligrams a day to try to get their blood Phes sort of tailored to where they needed to be. So again, they got randomized to either 20 or 40, and we had some period of time to be able to get them ramped up to that dose and had some flexibility in how fast we moved them up, and then they went through this placebo part of it. Our endpoints for that placebo part was, the primary one is the change in the blood Phe. So our hypothesis was that the people who went on the placebo, their blood Phes would go back up again, and the people who stayed on active drug, the blood Phes would stay down. The secondary endpoints were, really, does that make any difference to the patient? So we looked at scores of attention and mood, and our hypothesis is that the people who went on placebo is that their attention would get worse and their mood would get worse, and we'd be able to detect this. We only had a few people that were able to do this because we added it late, but we had 9 subjects who did a much more comprehensive battery of neurocognitive tests on the computer, and that also, our hypothesis is that those measures would worsen in the people on placebo. And this is what we did for mood. It was a POMS questionnaire, and then we had a rating scale of attention deficit, which the patient filled out with a neuropsychologist, basically looking at symptoms of their attention. And then this computerized test, and these were all tests that we knew in some publications were found to have changes in the outcomes in people with high blood Phe, so that's why they were chosen. So then people again moved into an open label extension, which has been out to 212 treatment weeks now in some individuals, is how long that they've continued on treatment, and we've continued to follow things like mood and attention. That's that. So again, I already mentioned it was flexible dosing up to 60 milligrams. So this is the characteristics of the patients that entered into PRISM 1. So we actually had, if you can do the math, it's 261 subjects that entered the trial. And the key thing to point out is that most of them were taking quite a large amount of protein. The daily protein intake is quite high, so they really weren't restricting very much, and their baseline blood Phe levels were quite elevated, obviously. And their inattention scores were quite significant. So there's not really a norm here, but the attention scores, the psychiatrists tell us if they're over 12, those are the people that they consider putting on stimulants for an attention deficit type of drug. And there was a large set of subjects in that group that actually did have quite significant inattention. So as I mentioned, that's the study start, and I can't give you the punchline, but I'll just say it's interesting data. Let's just say that. So the website at BioMarin has a press release that you can read, and it's got the detailed information about what happened with the Phes and what happened with the attention scores, et cetera. If you want more information about the trial itself, you can go to this website, clinicaltrials.gov, which has just the basic study design associated with it. So I wish I could give you more, but that's what I could say. Anyway, we're very close. They're working on the paperwork. The plan is to get everything to the FDA hopefully by the end of the year, and then we have to wait and see what will come of that. The process is that there will be another committee at the FDA convened independent of the one that we've been working with to take a fresh look at the data, and it's at that point actually that parents and well, in this case it'll be individuals, adults on the drug that will be extremely important in being able to have their input into the FDA about their experience with the drug that hopefully will help us get this over the hurdle and get it approved.